I'm particularly proud of this title, and I'm really pleased to be here to give the talk. I have a history with cancer pathways from back when it was Gilda's Club. I love the place, so I'm really honored to be back here. I've given talks here before, long ago. We were just talking about how I helped judge the literary competition, which was incredibly inspiring. So how we know what we think we know, that's kind of statistician talk, because being a statistician is never having to say you're certain. There's always doubt. But we learn things, we make progress, and here's how we do that. So start with the definition. Not sure that shows up in the back. Clinical trial is a prospective study, organized experiment of therapy in a defined patient population. We're not talking about observational studies. We're not talking about looking at patients' charts and trying to make sense of patterns. We're talking about an experiment that goes on in the human population, planned, conducted, and analyzed. Here's a cycle of a clinical trial, if you will. Does that pointer show up? Starting with a concept, an idea, just a seed, which proceeds to study design, and then protocol development. The protocol is the master plan for how the study will be conducted, how patients are treated, how data are gathered, how data are analyzed. Study build I have here, that's stat center talk. Once the protocol is ready, then people in the background develop the data forms, develop the database, and get the study ready for implementation. Then we go to activation and conduct. Data come, in case of our trials, in my organization, they come from all over the country and all over the world. Data are submitted now electronically. When I started, it was on paper. And we manage the data, solve the mysteries. There's always something missing or something inconsistent. So we go back and forth with the institution to make sure the data are complete and correct as possible. Then the analysis, reporting, dissemination of results, and those results then generate new ideas. Here's another look at that cycle. This one's going to go counterclockwise. A research question involves what population do you want to study? What phenomena are you interested in? Study plan, there's the protocol. Implementation, study build, study conduct, findings in the study. And then here it says infer. So you do a study, you have the results, you look at the data, and part of the statistician's job is to try to make sense and make the best conclusions possible from the data that were gathered. That I've labeled here truth in the study. This is what the study showed. This is what we think we know from the study. But then there's a final leap of faith, really. I can't say it any better than that. What is the truth in the universe? We have our study population. These are the results. We assume they apply to the general population. And in the experience that I've had, most of the time they do. But part of what statisticians do is quantify how often those results actually do generalize. So just a little bit about how clinicians and statisticians come together. So your clinicians of your person you're caretaking for, or you, is really interested in that person right in front of him or her. What do I know about this disease and this person? What are the best treatments available? Trying to make a decision that's best for that individual. Whereas statisticians look at groups of individuals or samples of individuals that are in a trial and make judgments then about the population of patients more generally. And we then make these inferences in the presence of uncertainty. So we meet together in this research reasoning universe, clinical and statistical reasoning. Again, develop a concept together, design and establish the framework for the study, conduct the study, collect and analyze the data, draw conclusions, draw inferences, and disseminate, importantly, disseminate the findings so they're available to the general public. So we do our best to have a good design, one that isolates the variable of interest, which would be a difference in treatment, and takes account of all the other possibilities that might explain the results. And in the final stages of clinical trials, we randomize patients to make sure all the other possible, what we call confounders, are balanced out so that they're similar men and women on both arms, similar age groups on both arms. So the results, if they're positive, for example, are due to the intervention that we applied and not due to some extraneous reason. A poor design can't really be rescued by statisticians, although there's a cottage industry of statisticians who try. But if you've got the wrong population, if you didn't measure the outcome well, you didn't plan well, there's no amount of statistical analysis that can really rescue. One of my colleagues often described this as you get more and more patients, but if you're measuring the wrong thing, you're more and more precise about the wrong conclusion. Good design, then, you have the outcome of interest, often survival time, sometimes shorter term endpoints like progression-free survival, time without increase of disease. We need something that's measured directly. We want the study population in our trial to represent the population of interest that we're going to generalize the results to. Importantly, we want the trial to be feasible. We want the intervention actually to be doable. We don't want something that patients stop taking the drug because it's too toxic or too inconvenient. So importantly, the intervention needs to be one that's adhered to. And of course, the design needs to fit the patient population that we're interested in, the disease and the stage of disease and so on. So here's a quick outline of phases of clinical trials. A new agent, for example, on the left, undergoes lots of laboratory testing. We call that preclinical, before the human experience. I've got on the bottom of this a rough timeline of how long this can typically take. So preclinical, animals and test tubes, a year or two. This is often done in industry by the major pharmaceuticals or the younger biotechs, sometimes in university laboratories. But eventually, someone is interested enough of a new compound to actually file what's called an investigational new drug application, or IND, and that goes to the Food and Drug Administration, and they judge whether the preclinical evidence is sufficient to move that compound, that new therapy, into human testing. So once there is an IND, then you can try this on humans. Phase 1, and I'll do this a couple times, but phase 1 is typically a dose finding or safety study, phase 1 clinical trial, in small numbers of patients, 20, 30, 40, trying to find the appropriate dose. So it's highly experimental, and we can talk more about that when we're done. So once a dose is found that's thought to be effective and is not too toxic, then that often moves into what we call phase 2. In phase 2, we do a larger trial on humans and try to get some efficacy information. We think we have the right dose. Does it seem to work? Does the tumor shrink? Do patients seem to live a little longer? Just enough evidence that we're confident that now we're ready for a big phase 3 trial. In phase 3, then, we typically do a randomized comparison of new therapy versus standard, or standard plus new versus standard, A versus B, or A plus B versus B. And those are typically in the hundreds and sometimes even thousands of patients. To do a comparative trial, is the new really better than standard? A little more graphically, early phase 1 safety and dose finding in a handful of patients. Phase 2 looking for hints of efficacy. Does it seem promising enough to do a major phase 3 trial? And then phase 3, the definitive randomized comparative study. So phase 1 has no control group. Actually, I don't know if there are people that are involved in other fields of research besides cancer, but in heart disease or diabetes, a new agent will come along and it's typically not terrifically toxic. In phase 1 trials in most diseases outside of cancer are done in human volunteers. We try it and see if it's not too toxic for normal volunteers. In cancer, these are done in patients because typically cancer drugs are highly toxic. Cancer is a pretty serious disease, and the agents are not perfect. They always have some toxicity. So phase 1 are done in cancer patients, starting with a low dose, and if it doesn't seem too toxic, the next group of patients, a higher dose, but there's no control group. It's just escalating doses until you find something that's not too toxic and it might have some activity. Phase 2, the reason I'm warming up to that, phase 2 is more complicated. There are randomized phase 2 trials and there are single arm phase 2 trials. So it depends. If it's a disease where there's been no progress for a lot of years, you kind of know what the typical response is, then phase 2 might be without a control group. But if there's more variability, maybe you want a randomized control group, even if it's not a huge definitive study, just to get a hint that maybe it's better than the standard. So phase 2 can be randomized or single arm, and phase 3 is always randomized. So if the informed consent process is done correctly, it should be clear to the patient what's going on. In phase 1, I often wonder about that consent process and how truly informed patients are. It's a difficult conversation. But in phase 1, we're not really sure that the agent is effective. And patients should know, maybe it's their last best hope, but there's not yet a lot of evidence that the new thing is really going to work. And to me, there's a lot of altruism that goes into participation in a phase 1 study, because I would hope that they're not oversold. This is the greatest, best thing, and you're going to be cured, because that's not usually the case. Yeah.
In the control group, is that no treatment or standard course of treatment?
In cancer, it's usually standard treatment. Okay. There's almost no case I can think of, except maybe after surgery, when the cancer is gone. You're talking about observation then versus some intervention. But typically it would be standard of therapy. Yeah.
He answered one of my questions, but then I came up with another question. Since then, would you say most patients find these trials on their own, or is their doctor recommending them and finding them? How do they learn about That might be somewhere else later.
Yeah.
Like if I was a clinician, then I would hopefully share all the risks that are coming along.
Yeah.
Curious, like where they find out about the trial.
So, yeah, so it varies. I mean, some patients, some people are really good at doing the research themselves, and they come to their physician, say, what about this? Others are more passive, and you hope then you have a good oncologist who knows the field and knows what trials are available. And that's usually the case.
Out there that really talk about like phase 4 clinical trials. Here's these options. It's really a lot of social networking, going out there, doing your Google alerts, on daily, all these different things that you want to kind of narrow down things, and then continuing. Is that for me? No.
Yeah.
And then go back. That's how I.
Yeah, I would hope most people are like that, but most people aren't. I mean, not all people are that. Some people are pretty passive about it and rely totally on their doctor. I would urge patients and their caretakers to urge their patients, their friends and relatives to do research themselves. Right.
How would you explain the combined phase 1 trials?
Combined phase 1 and 2. So there are such things, as you probably know, probably for reasons of convenience. It takes so much time and effort to get a protocol together. Sometimes you package a protocol into something called, say, phase 1/2. And there would be a part of that protocol in which there's dose finding. And then once you find a dose using the same protocol, everything's laid out, then you move into a larger study in the determined dose. That can be an efficient way to proceed. So does that answer your question?
Yes, I guess I was curious to know your phase 1. I was a bit afraid that it's not like there's some outbreaks. So what about the combined like phase 1?
So in terms of consent, for example, a proper consent would tell the patient the data up to then. Okay, now we're in phase 2 part of this protocol. Would you agree to participate? So that should be part of a consent. Good. Any others? Yeah. So there's an elaborate set of studies that are done in preclinical, including test tube. But still, often the penultimate stage is a rodent. And you look at toxicity in the rodents, and you find it's going to be a little. It's called the lethal dose 10. So a dose that kills 10% of the mice, for example. 1/10 of that dose would be the starting dose in humans. And that's based on a lot of experience. That's the kind of thinking that goes into it, including cell cultures. Okay. So these are some of the things that we think about in designing the protocol. What really are the objectives? What are we trying to do? What endpoints are we measuring? What is the right population for this new agent or new regimen? What are we trying to prove? How big a difference are we looking for? How much of an improvement? Those things lead to a conversation about how many patients we need, which then leads into how long the study takes to accrue. And then finally, in phase 3, we talked about the randomization strategy. So this is really textbook stuff about Stat 101. We really do this in cancer clinical trials. The objective, to draw a conclusion about the effect of a treatment in a population of patients, that's everybody, using information from the sample of patients, that's just those who are in our trial. We set up a null hypothesis, a straw man that we're trying to disprove, that for example, the new treatment is the same or no better than standard, against an alternative hypothesis that we're hoping to establish, that the new treatment is better than the standard. And here's a key for statisticians: new by a specified amount. Otherwise, we can't make progress about how many patients are needed to establish the result. That specified amount, whether it's 3 months more survival or 10% greater response rate, should be something like the minimum difference that's really clinically meaningful, but it also needs to be achievable. So there's a little bit of a dialogue that goes on with the clinician and the statistician. And we finally arrive at a sample size that helps distinguish between this null hypothesis of no difference and an alternative of a specified difference as the true state of nature. Endpoints I talked about, there's a lot of them to measure in a cancer clinical trial. As you know, things about toxicity, things about the response of the tumor to the drug, does it shrink or not, a specified amount? What is the length of time before the disease gets worse, or the length of time before relapse? And overall survival, still very important endpoint. Yeah.
All of the trials work, or is there a point out of the trial?
So a patient should and can always withdraw from a trial if he or she thinks they're not receiving benefit. And that's understood. It may be disappointing to the oncologist, but it's not their job to adhere to the protocol over the needs of the patient.
Toxicity, is that usually the main why a drug not progress?
It's often the case that a drug is too toxic and limit on toxicity is reached before any benefit happens. So drugs can fail at phase 1. More often you find a dose that you think might be effective, but then it proves not to be better than standard. So I'm going to end this with an upbeat. So for 20 or 30 years of my career, 90% of the trials that I got involved with were negative. Past phase 1, maybe even past phase 2, but randomized trials showed little or no benefit. Last 10 years, there's been a lot of progress, a lot more positive trials, but with less toxicity. Yeah.
There also might have been an implicit question that she was asking, which is if the clinicians are not diligent about, I mean, the clinicians can cherry pick if they aren't doing it right. They can exclude people that then aren't considered in the statistics, which can give a wrong impression. So you talked to about that.
Yeah, that's a great point. And statisticians kind of achieved a consensus 30 or 40 years ago. So suppose you're doing a randomized trial and the new regimen is looking a little toxic, and patients start dropping out because they're getting sick and they can't take it, or maybe they aren't getting a benefit. How do you compare that treatment arm with the standard arm? And the statistical response, and we finally convinced our clinical colleagues, everybody who's randomized is analyzed in the arm to which they're randomized. It's the only unbiased way to compare. And if patients drop out, then it just is an indication that the treatment is not working. It's not being adhered to.
So good clinicians shouldn't knock people out of the statistical analysis.
Absolutely right.
They do their considered though.
That's, he's saying that's the right way to do it.
Yeah.
Yes,
Yeah.
absolutely.
We call that intent to treat. Fundamental clinical trials principle. Thanks. More about endpoints. I think I'll move ahead past that. In cancer, we have these standards now, which actually didn't exist when I started. So it's nice to have good common definitions of toxicity. The US NCI has what they call a common toxicity criteria. Side effects are graded from a 0 to 5 scale on how severe they are. 0 is nothing. I won't tell you what 5 is. A key endpoint, especially in early clinical trials, we call response. And it has to do with, does the tumor shrink up a specified amount? 50% by area, it turns out. Yes or no. It can come in a more graded system where complete response would be disappearance of the tumor. Partial response would be shrinking by 50% in area. Stable disease means it's not getting worse, and then progression. Most of our trials, when we get to phase 3, are talking about a time to endpoint, either time to relapse, time to recurrence, time to worsening of disease, or time to death. Yes. Yeah, you'd think, wouldn't you? So it's kind of a carryover, and maybe it'll improve and change. It's a carryover from the early days of X-rays, looking at plain film. There have been a lot of effort to improve the definitions, but so far it's more work and not much advance. So we've sort of stuck with that definition. This is the most technical slide I'm going to do, I promise. But it has to do with that inference that we make at the end of a trial. So in this paradigm, where we set up a null hypothesis and alternative hypothesis and try to decide between the null and the alternative based on the results of the trial, all we know is what's in the trial. We don't know what would happen if we apply this to everybody, to the population. So I've called that latter thing truth in this slide, and I've called the former thing the conclusion is based on the trial. So sad fact is, for regimens that are not active, for example, we could have a negative trial. That's the right decision, it turns out. But we could also, in our trial, with an inactive agent, conclude that it's active. And that's a mistake. So if it's inactive agent and we say that the regimen is not active in our trial, but it's active in the population, that's a mistake statisticians call a type 2 error. You could also think of it as a false negative. The trial is negative, but the truth is different. Conversely, if the regimen is active and we conclude no difference in our trial, that's a false positive. We say it's active, but it really isn't. What we want to do when it's active is conclude that it's active. So there are these two kinds of mistakes, false positive and false negative. And the statistician's job is to quantify those errors. And we design a trial so it has a small probability of a false positive and a small probability of a false negative for the null versus a specified alternative, and that drives the sample size. How small? There's another sort of arbitrary thing. We for reasons that are not particularly good, but have held up over time, we say that the false positive rate shouldn't be more than about 5%. So we're willing to say that 5% of the time when we say something is active and it really isn't, that's the price of doing business. 5% false positives and 10 or 20% false negatives. I can't justify it any better. We know that they should be small. That's the convention that's been established. So you go to the FDA and you do your trial, they'll want to know that the design is such that the false positive rate's less than 5%. They don't really care about the false negative, but you do. So you want that to be small too. That's as complicated as I'm going to get. Just a ballpark, let this sink in a little bit. If you have a trial where the outcome is time to something, which most of our phase 3 trials are, it's not the number of patients that's important, it's the number of events. So in a trial in advanced disease, stage 4 colon cancer, stage 4 lung cancer, events happen in a hurry. So the number of events is pretty much the number of patients. And a typical phase 3 trial in the advanced disease setting would take a couple hundred patients. If you're talking about breast cancer, after surgery, basically cured, we think, almost. Yeah, so suppose the primary endpoint is survival. So the event is death. So we're looking to time to death. With advanced disease, patients die in 6 months, 12 months. With breast cancer, after surgery, patients have virtually normal survival. So you get thousands of patients for a couple hundred events. So that's why the breast cancer trials take many more patients than a lung cancer trial. Just a little bit to wrap about the structure that we use for clinical trials in the US. It's a long history. National Cancer Institute goes back to the 30s. It was the first of the NIs of H, first of Institute of NIH, because there was no NIH then. Got subsumed in the 40s into a set of institutes that we now know as NIH. In the 50s is when we first started doing clinical trials in the US in cancer, sponsored by the National Cancer Institute. Since the 50s, we've had a system funded by the government through the National Cancer Institute of standing groups, oncology groups, consortia. There's a long history of it, but right now there are 5 major groups in the US that are national in scope that study cancers. 4 of them study adults. They're now called the Alliance, NRG, which has a history. It's breast cancer, radiotherapy, and gynecology. ECOG, which started out as the Eastern Cooperative Oncology Group, and it hyphenated with a radiology group. And SWOG, which now is just SWOG, which is hard to even say. But it used to mean the Southwest Oncology Group. And in the 50s, there really was a regional basis for these networks. Now they're all national. So there are 4 studying adult cancers, and there's 1 children's group, which merged from 2 about 10 years ago, called the Children's Oncology Group. Those are the networks in the US studying cancer. Mission is to improve lives through cancer clinical trials and translational research, bringing in some basic science. As you can see, SWOG is a national organization. You might notice this strange lacuna in the central part of the US. That's really mostly due to the Mayo Clinic and its network, which is part of the Eastern Cooperative Oncology Group. That's why SWOG doesn't have much participation there. But otherwise, pretty much a national group. There is a set of offices that comprise the leadership. Here in Seattle, we have the Stat Center, as Glenn mentioned, co-located at this company called CRAB, which does data management primarily, and the Fred Hutch, where the statisticians by and large reside. Two organizations that work together to do the statistics and data management. Our group chair is now at UC Davis. Actually, we have two chairs, one at UC Davis and one in the Northeast at Columbia University. We have a philanthropic foundation based now in Michigan. And we have some folks that put together the protocols, and they're based in San Antonio. So this has evolved over time. Used to be all one big office, and each time we compete for a new grant, we kind of come up with a different way of doing business, but that's how it is now. 60 some years, we've done over 1,300 trials, enrolled a couple 100,000 patients on our clinical trials. We've done studies that lead to 15 drug approvals, and we've done studies that led to 2 drugs getting taken off the market because the initial positive results turned out to be false positives. Over 100 changes to standard of care. We have a biobank of nearly 1 million samples, so a lot of research goes on with that. And we have a clinical trial database here in Seattle. It's not yet in the cloud. It's actually physically on the 7th floor on Westlake with all that trial information. And we did a little calculation a while back and published this result, which we're pretty proud of. If take our positive trials and assume they get disseminated, which we hope they do, and improve the lives of cancer patients, we estimate that we have extended 3.3 million years of life at a cost of about $125 per life year saved. So that's pretty good.
Yeah, we were just talking to this. He's wrapping up, and then we're going to go to this question. Thank you.
Okay, so I'm going to do one little diversion, and then we'll do what Glenn wants. Son, my long lost son. So this is my project that I'm still involved with. Somebody mentioned my age, so I say I'm semi-retired. But I'm still involved in this project, which is disseminating our network, which I've said was national, into the hemisphere. So we have what we call the SWOG Latin American Initiative. Been working on this for 15 or so years. Harder than I thought it would be. What I found out in travel in the region, that countries to the south of us are incredibly varied. This just, the percent indigenous people is what's being shown in this graph, just an illustration of how different each country is in Latin America, with a high percent indigenous people in Guatemala, Bolivia, pretty high in Peru and Ecuador. And there's one little white thing here in Uruguay, which when we visited, I thought I was in Italy. I mean, there are no people of color in Uruguay. Among other things, there are different patterns of cancer in Latin America. So this is showing rates of stomach cancer for females, turns out, but similar for males, with red being a hot spot. So there's high rates of stomach cancer, especially along the Andes in Latin America. It's due, in fact, to a bug called Helicobacter pylori, which is endemic in Latin America, exists to some extent here as well, treatable by bacteria, by antibiotics. But it's endemic and it causes stomach cancer. And it's been around since, at least since these mummies were pulled out of the Andes 10,000 years ago. We've done trials of eradication. What's the best way to get rid of the bug? And there's now an international trial going on testing whether if you get rid of H. pylori, do you reduce the incidence of stomach cancer in future years. So that's kind of fun sidelight here. The sites we have the major cancer centers in Mexico, Peru, Colombia, Uruguay, and Chile in our group. And I've been privileged to travel, both of us have. And I was particularly on one trip enamored by a town called Valparaiso, just to the west of Santiago on the coast. It reminded me, I'm showing my age, Haight-Ashbury in the 60s and 70s. And the street art was incredible. It says, "We are not hippies, we are happies." So that's been a lot of fun. Questions?
I think one of the most important parts for me in the work that I do with, questions about clinical trials is it's very, very difficult for people to understand anything about them, really. And when I was doing clinical advocacy work, I had patients who were oncologists asking me to help navigate to find clinical trials for them through clinicaltrials.gov, which is a very confusing website if that isn't the language you speak. So I say that because it's tricky because it's such a precise or robust science that I think the layperson who's just diagnosed with cancer really can't comprehend any of this. It's already difficult to comprehend the most basic things after you're diagnosed. So the first question, someone wrote in and said, if someone is newly diagnosed, at what point should they start thinking about clinical trials?
Right then.
Okay.
Right then.
Okay.
Chances are there's a trial for just about every kind of patient at every phase of their treatment. I'm a believer in cancer clinical trials. Obviously, it's what I've done with my career. You get at least standard of care treatment. You get lots of extra attention for being on a trial. Our article of faith is patients on trial do better than patients off trial.
That's great. It's really great to know because this is information that I think a lot of people don't know. And to follow up with that, when you talk about like at least getting the standard of care is such an important part because the pushback I've gotten from our members in groups is, well, I don't want to do a trial because I don't want to get the placebo, right? And I try to explain to them when it comes to oncological trials that it is standard of care. Is that correct? It's like standard of care or standard of care plus the experimental arm. Sorry. Okay.
Yeah. There are very few placebo-controlled trials in cancer. I mentioned before there might be some where cancer is excised completely at surgery, and standard of care would be observation. And those might be placebo-controlled for reasons of adherence.
Gotcha.
That's rare.
Yeah, it's so good to hear that because there's so much resistance about clinical trials, and then the other side of that in our member population is they sort of equate clinical trials with a Hail Mary, that it's when nothing else works, and I know myself that that's not the case, but I'd love to hear from you.
So we talked about phase one. There's some Hail Mary-ish parts of these phase one trials. It's a situation where you may have been through several lines of therapy before, maybe several trials. There's nothing else available that's commercially available. There's a new agent that is being tested. It's a hope. It's also a contribution you can make by participating.
Right. Okay, so here's one. What are the costs? This is several questions, but what are the costs associated with clinical trials to the patient? Like they're asking, how much will it cost me to for travel, lodging, etc., and are there resources to help with those costs?
Yeah, that's really a great question. So in the government system that I participated in, the costs of the treatment are covered by the trial. They may be referred to insurance or whatever the patient brings, but any extra cost for the trial, the treatments on the trial are covered by the trial. Transportation, lodging are not covered by the trial. That's an important issue. Many institutions try to supplement by covering those costs. Some do and some don't. It's not part of our trial reimbursement.
I see.
But for equity, it's a really important point.
What do you think is sort of the best, like if we had a blank check, what would be the best way to sort of engage certain communities to increase, mortality rates, I guess?
So let me talk around that.
All
Maybe
right.
I won't get right to the
Sure.
point, but one of the good things about a consortium like ours is that we have not only the research institutions like the Hutch, Swedish Virginia Mason here in town, but we have community centers. So the trials that we do, those hundreds of trials, are available throughout the community-based centers as well. That's the good news. We've done a fair amount of research into barriers for entry into clinical trials. And primary barrier turns out to be the oncologist. Maybe they just don't tell the patient about the trial. So that gets back to one of the early questions. You got to be your own good advocate. Look at this. Primary barrier is the oncologist not presenting trial to patients. Second is the fear of financial toxicity, which would be transportation, lodging, and so on. So if there were a magic bullet, it would be to try to cover those costs to make sure things are equitable.
Okay.
We have good data that once a patient enters our trial, things like socioeconomic status, race, income don't matter, but they matter getting into the trial.
Gotcha. And that's a problem.
Yeah.
Yeah.
That's a problem if you think trials are good, which I do.
The question is, what is the process of compassionate care access to trials if you don't technically qualify for one, but are out of options?
So probably there are people here that know more about this than I do. If so, speak up. But there is a system called compassionate use or compassionate care where the IND has been approved, so the drug is available at least in certain conditions, but maybe you don't fit the trial for that drug. Your physician can appeal on a case-by-case basis to the FDA to make a case for using that drug in that patient. I don't know how successful that is. It probably depends on how much power and influence you have.
Yeah, which again gets to the inequity issue, say the least. Okay, so this one says, my doctor said there were no clinical trials available when I asked, but I then found one in California that I am now a part of. Why is that?
So you got to be your own advocate. Maybe an oncologist doesn't think they get reimbursed enough for the time and trouble. Maybe they're not totally dedicated to trials like I am.
I think, well, I'll just close with this. I think the question that was asked that you answered about if someone's newly diagnosed, at what point should they start a clinical trial? And you say right now, like, and I feel like that's something, there needs to be a tremendous amount of education on on that, because I just don't think people know that. And people who are pretty, you know, astute and plugged in.
Yeah.
But so the question becomes like, are there things in place now that educate patients about, you know, like what you just presented, right? But in a way that someone could watch a 10-minute video and go, oh, I didn't even realize it. I thought it was just like if you're going into hospice, you have these things. But to your point, like the efficacy, the potential.
Yeah. I don't know the answer to that.
Okay.
There probably are with the vast internet out there.
Yeah.
Just don't know.
Okay. But you're not doing it at Crab, and why not? I like that.
Because I'm shy.
Okay. That's fair.
So-
That's fair.
Is there any patient demographics that you would say pursuing or looking into is not right? Or should everybody go look at a clinical practice? Like, would you tell anyone no? Don't bother.
I would not tell anyone no. So I've got a little story. It's more about diagnosis than treatment, but I think it generalizes. I have a couple of friends. It's about being your own advocate. A friend who wasn't feeling so good, a little anemic, not sure why. And I said, get your protein tested. And he had myeloma. The local doc should diagnose that. I shouldn't have to diagnose that. Another friend of ours, local, had a little spike in calcium. Doctor was ignoring it. Said, pursue this. Why is that? And she turned out to have myeloma. So it's just a lesson that you're your best advocate.
More importantly, I just want to say thank you so much for taking the time to.
Thank you. My pleasure.
And
And for all of your work over the last so many years.
thank you.
Thank
guys.
you all. Nice to meet you.